Hello everyone. This presentation highlights the various applications of the SCIP database and its utility in advancing fundamental knowledge of plant defense against combined stress. The presentation also shows how the SCIP database provides novel insights into plant stress tolerance mechanisms under complex environmental conditions. The SCIP database comprehends the research on the impact of various stress combinations on plants covered in 734 publications on 122 stress combinations, which show the study on 325 organisms, including plants, pests, pathogens, and the insect vectors. The SCIP database also hosts information on 29 transcriptome analysis under the combined stress conditions, and also gives an account of 107 traits affected by the various stress combinations in plants. The information from the SCIP database can be utilized for both the fundamental and the applied research. The database is a compendium of research on stress combinations in plants, and it highlights complex environmental conditions that affect plants, which is covered under the abiotic sections of the SCIP Phenomics. It also highlights the impact of abiotic stresses on plant diseases, which is covered in the abiotic section of the SCIP Phenomics. And further, it gives information on the existing disease complexes which impact plant growth, which is covered in the biotic section of the SCIP Phenomics. Further, the SCIP database also serves as an information resource on the crop improvement, and it highlights the possible candidates that can be used for crop improvement under the stress combinations. It also gives an account of the various plant traits affected by the stress combinations. As a contribution to enriching the knowledge on fundamental research, the comprehensive literature information in the SCIP database helps in providing answer to few of the top 10 unanswered questions in the MPMI. For example, the very pertinent question posed on the MPMI platform on how does abiotic stress such as climate change influence plant microbe interactions is answered in the SCIP database. The SCIP database, through the literature information compiled, provides a comprehensive account on the effect of important abiotic stress conditions uh, and their effect on the various plant diseases. The analysis highlights important abiotic stress factors like drought that significantly aggravate many plant diseases, and also indicates the abiotic stress conditions like temperature and salinity, which also play a role in reducing some of the intensity of some of the plant diseases. Thus, the database may indicate possible disease management strategies and also avenues for research in this regard. The database also provides insight into another important question posed on the top 10 unanswered questions in the MPMI on how does microbe-microbe interaction affect plant microbe interactions. The biotic category of the SCIP Phenomics provides an account of the various interactions between the different pathogens and pests and the impact of such interactions on the plants, thereby highlighting the existing and or emerging disease complexes. Here in this diagram, which has come from the analysis of the SCIP database, we can see that there are various pathogen interactions which have a negative impact on the plants, shown by the red arrows, whereas the green connecting lines show that such interaction between the pathogens or the pest have a positive impact on the plants. Furthermore, the SCIP database also provides broader account of the various stress combination affecting economically important crops. Thus providing a platform wherein we can visualize the various stress combinations posing a threat to agriculture. For example, herein we see the various stress combination affecting legumes and the plants which are majorly affected by the stress combinations. Thus, the SCIP database provides an account of the current research that is going on and also the avenues for further research in the field of stress combinations. Furthermore, we can see from the example of the studies compiled on rice that that among the various stress combinations, biotic stress combination is the most evident, affecting the rice production, whereas the abiotic stress combination is the most damaging. Herein we can see that all the abiotic stress combinations have negative impact on rice crops. The SCIP database also provides an account of the global impact of stress combination on plants, indicating the various hotspots of research on various stress combinations. For example, the database provides a glimpse of the places where the plant diseases are aggravated by the prevailing environmental conditions. Also, SCIP database highlights the places where complex environmental conditions are posing threat to the agriculture. Moreover, the SCIP database also gives an indirect evidence on the prevailing and the emerging disease complexes negatively affecting the plants at various places around the globe. Furthermore, from the analysis of research on various stress combinations, the SCIP database provides an account of major plant traits that are affected by the stress combinations. This advancement in fundamental research can be utilized by various tools for simulation modeling, opening avenues for further applications in the applied research. For example, we are using the information collected from the SCIP database. We are developing a deep learning model to predict the impact of stress combinations on the plants. Further, the information on SCIP database can be utilized for applied research as well. For example, the SCIP database transcriptomics sections can be utilized for answering yet another question posed in the top 10 unanswered questions in MPMI on how to translate basic research into emerging crop plans. The SCIP Transcriptomics is a platform where all the Arabidopsis thaliana transcriptomic datasets are available and can be accessed. The meta-transcriptomic platform compares the transcriptomes of Arabidopsis thaliana under the various combined stress conditions, and it provides a list of common and or unique genes to the stress combinations. So we have chosen two genes from this platform, namely the ProDH and SARD1 and CBP60 gene. And we have validated the role of these two genes under various combined stress conditions. For example, the function of ProDH was validated under drought and Ascochyta arabi infection in chickpea, whereas the function of SARD1 and CBP60 was validated under drought and Pseudomonas syringae infection in Arabidopsis. So as a case study 1, we try to find the role of ProDH under the combined stress condition, combined drought and pathogen infection in chickpea. So from some preliminary analysis, we knew that ProDH has a role to play in plant defense against the stress conditions. So we looked for the presence of this gene in the combined stress transcriptomic dataset. So the genes can be searched here in the search tab by writing the name of the genes or the gene IDs. So we could see here that proline dehydrogenase is involved in plant defense against a variety of stress combinations, both the abiotic stress combinations and the biotic stress combination. So moving ahead, we probed into the function of this gene using functional genomic approach. We silenced ProDH2 and the transformed silence plant showed that the gene is involved in the enhancement of chickpeas defense to Ascochyta ribeye under the drought stress conditions. Thus, using the leads from the database, we could unravel one important mechanism of plant defense against drought and Ascochyta under combined drought and Ascochyta infection in chickpea. We know that drought leads to proline accumulation, and proline is catabolized into P5C, and P5C has a role in pathogen defense against the pathogen infection, although not shown in chickpea. So in case of combined drought and pathogen infection, we have shown that the catabolism of proline to P5C via ProDH2 has an important role in enhancing the defense of plants against Ascochyta ribeye infection. So the study, preprint of the study is available at bioRxiv. So as the second case study, we chose the genes SARD1 and CBP60. So the role of these genes was under the combined stress conditions was looked into using the database. And we found that SARD1 is involved in plant defense against drought and botrytis cinerea infection in Arabidopsis. So to validate its function in drought and Pseudomonas infection in Arabidopsis, we have first of all looked for the expression of the genes in Arabidopsis under the combined stress conditions, and then the gene was downregulated, and the results showed that the two genes are major determinants of disease outcome under the combined stress. So here, and also we could find a mechanism of defense against the combined drought and pathogen infection, taking the leads from the SCIP database. So it is known that drought leads to the activation of ABA signaling, whereas pathogen leads to the activation of SA signaling. And ABA is antagonistic to SA signaling. Then in the case of drought and combined drought and pathogen infection, the drought leads to inhibition of function of the gene CBP60 and SARD1, which leads to more pathogen infection under the drought stress condition. This study has been recently published. So furthermore, the SCIP database can also serve as an important resource for science communication and outreach. So the database hosts valuable research materials that can be utilized by various scientists and the students. This can be accessed from the download section of the database. Herein we see that various kinds of research materials are available, which includes the raw data files and the processed data files to analyze information on the combined stress conditions on all the plant species, and then the transcriptomic data sets, and then various academic teaching materials in the form of thesis published in the field of stress combinations in plants, posters, various protocols for imposition of the combined stress, and the database also provides a list of important labs working in the field of combined stress studies. So it can be accessed. The whole list can be downloaded from here. Also, the database hosts several videos that have information on the stress combinations and also the concepts of stress combinations in plants. For a researcher, thus, the SCIP database provides the insights into the various new avenues for research, and also it provides an overview of the significance of the field of research and the overview of the current research which is going on. SCIP database can also serve as an important resource for agricultural scientists to gain information on areas which can be explored for better management of crop diseases. Thus, we believe that the SCIP database has multiple applications and tremendous utility and can be a very useful resource for the plant research community for both expanding knowledge on fundamental research and providing insights into unexplored area of research in the field of stress combinations, as well as it can be a valuable resource for identification of novel genes for crop improvement in the under the complex environmental conditions. I would like to acknowledge the efforts of all those who who worked hard for the SCIP database. So the team includes lab alumni and current members working on the database. Also, the SKM lab team for the support and efforts for the validation studies, and the funding from NIPJAR and the other agencies. Thank you.